So the first half of the course, we concentrated on structures of microbes, right? We looked at capsules and cell walls and shape. Now we're starting to focus more on how the microbes function. What can they do? Right? So structure and function. It's almost analogous to anatomy and physiology, right? For multicellular organisms. So again, does an organism have a capsule? That's a structural component. Of course, it does have some functionality to it. A big question that we ask a lot, especially today as we were doing these tests and looking at our results from the different media that you've employed, is fermentation, right? Can they ferment different sugars? Glucose is one of the sugars that we look at. So you guys ran the test, methyl red and Voges-Proskauer. We've already recorded our results for that, but we're going to talk about, okay, well, what does this mean, right? What does this tell us about our organisms? You have your Phenol Red Broth tubes that you had already inoculated. You've been looking at and recording those results, and then we're going to discuss what do they mean. Same thing with your citrate tubes. You've been looking at and recording your results. We performed the Catabolism test already, and then we're going to talk briefly about Anaerobic. These test kits are about $20 a piece, so budget-wise, we've eliminated them from the lab. And I don't even have some old samples to show you guys because of the loss of my refrigerator. I lost most of my old stuff. But I've got some pictures. There's pictures in your lab manual, and you can see how some of these tests are performed in, say, a clinical lab setting. They wouldn't necessarily do a whole bunch of individual tests like we did. They use these test kits that will do a whole bunch of tests all in one strip. So they're pretty cool. And they're called an Anaerobic, and the particular one that we looked at was Anaerobic 2. So for that particular grouping of organisms, it would help you differentiate which one you got. So these are all differential tests, right, where we can differentiate one aspect from another, one group from another, and based on what they can do, mostly biochemically. Can they ferment? Right? Can they utilize different sources? So, basic redox reactions, chemistry, right? These are chemical reactions, right? So the best saying that always works for me is oil rig, right? Oxidation is the loss of electrons, reduction is the gain of electrons. And these usually go hand in hand. Right? Somebody loses the electrons, someone else picks them up, right? So they're redox reactions. Reduction and oxidation typically happen together. So electrons don't just disappear, right? They go somewhere, right? So somebody takes it, gives it to someone else. In biochemical systems, in addition, protons typically move, or whole hydrogen atoms. Hydrogen is very important, right, for chemical reactions in biological systems like a cell. So in this case, these electrons and protons, these hydrogen atoms, are carried around by molecules in our cells. One of the most common molecular molecules, what is sometimes referred to as coenzymes, or very commonly referred to as electron carriers, you may have heard of, and it's an abbreviation for the full chemical name, which, God help me, I don't even think I can pronounce that one. But NAD is actually a modified nucleotide. So this is the nicotinamide portion, right, the N. This is the adenine portion of the nucleotide. So there's actually two nucleotides, right? These are what are in RNA and DNA, right? The phosphate group, the sugar, in this case an adenine. This is the same nucleotide you would find in DNA and RNA. This part, the second portion, has been modified. It has the nicotinamide on it. And this is what interacts with the hydrogen atoms. So in this case right here, it has a plus sign, right? It's lost electrons, or a hydrogen atom. And then it's going to gain it from an organic substance like malate, which turns to oxalate, which is one of the intermediates in the Krebs cycle, right, that we talked about last time. So this guy, and this is not showing the whole molecule, right? It's just showing where the change happens. So notice it gains a hydrogen, but essentially it's gaining electrons. It's carrying electrons. So it becomes NADH, because it, 2 actually. The 2 is cut off there, I'm noticing. And an extra proton is released, right? So this is a hydrogen atom without its electron. So it's basically just a proton. All hydrogen atoms are a proton and electron. They're the simplest of all atoms. So remember from last time, glycolysis, we can break down sugars, right, into pyruvate or pyruvic acid. Pyruvate is just the conjugase. A lot of instructors use these two terms interchangeably. And then it's modified into acetyl-CoA, and carbon dioxide is lost. And that acetyl-CoA enters into the Krebs cycle, or sometimes called the Tricarboxylic acid, which is what TCA stands for. Sometimes it's referred to as the citric acid cycle. It's one of the main components. And then electrons that are taken from and picked up by those electron carriers, like NAD, which becomes NADH, are going to travel to the electron transport chain, where they're going to give those electrons to that chain. Those electrons are then, if it's aerobic respiration, are picked up by what? Oxygen, and we form metabolic water. But anaerobic respiration, it's something else that's picking up those electrons. So in this case, it gives an example of hydrogen sulfate, I think this is, to form hydrogen sulfide in water. Ultimately, carbohydrates like glucose are oxidized, which means we're doing what? Removing electrons. And so the carbon is given off in the form of carbon dioxide. In the process, the free energy eventually is going to get trapped in another modified nucleotide, ATP. That nucleotide that has adenine, instead of one phosphate group, there are 3. That's what the T stands for, triphosphate. And so that stores that energy. When they break off that phosphate on the end, they release that energy again, right, for building molecules or doing other processes that require energy. So glucose is a beautiful 6-carbon sugar with hydrogen and oxygen. They're commonly referred to as carbohydrates because for each carbon molecule you have a molecule of water, right? It's carbon that's been hydrated. So again, we can take something like glucose, break it down into pyruvic acid. In the process, at the enzymatic level, we're going to generate ATP, right? We're going to add phosphates to ADP and store that energy for later. An electron carrier, NAD, will pick up. So it is NADH. Oh, I'm thinking of a different one where it's the 2. But anyways, picks up electrons, right? And it's going to carry those electrons to the electron transport chain. So again, it's a reduction reaction. It's gaining electrons, where the carbon is losing the electrons. The carbon molecule becomes oxidized. So this is the Embden-Meyerhof-Parnas, he usually gets left off, third person, pathway. There's another pathway that's similar, the Embden-Meyerhof pathway of glycolysis, and it produces less ATP, just one. It produces one NADH, but it also produces another electron carrier, NADPH. So just a little bit different way of, again, extracting energy from glucose. Pyruvic acid is converted before it can enter into the citric acid cycle into acetyl-CoA. Notice carbon dioxide is lost, and electron carrier NAD picks up some more electrons, and it will carry that later to the electron transport chain. So notice again, see how the carbon and the oxygen here is lost. The acetyl-CoA is then added to the molecule. Acetyl refers to this 2-carbon group. Here are your hydrogens, right, that end up over here. And there's your acetyl group, because it looks like acetic acid. So in the citric acid cycle, so now we have acetyl-CoA. It combines with the 4-carbon sugar known as oxalate. In this first step, the CoA is regenerated. Go pick up some more pyruvate. Hydrogen atoms are released. Citrate is formed, and that's why it's commonly referred to also as the citric acid cycle. Tricarboxylic acid is also another name for citrate. There are 3 carboxylic acidic groups on this molecule. So at the substrate level, some more energy can be transferred to the adding of a phosphate to a modified nucleotide. Typically it's GDP. It's just a different base on that nucleotide. Functions very similar to ATP. And then notice more picking up of electrons. NAD picks up electrons in another new electron carrier we hadn't talked about yet. Let FAD, this one picks up 2 hydrogens. More carbon dioxide is produced. Notice again, this is substrate level phosphorylation at the enzymatic level. Reducing, we're gaining electrons to these electron carriers. And carbon is oxidized. It has lost electrons. So the citrate test. That citrate sounds familiar, right? You guys looked at the citrate test. It detects that organisms can take up citrate from the media. Not whether it can synthesize it, whether it can take it in. The citrate also detects if they can metabolize ammonia. The first step in the citrate fermentation is essentially the first step of the Krebs cycle in reverse, right? So when they take in citrate, they use the enzymes, but they go in the opposite direction. They break it down for energy. In the electron transport chain, which is embedded within a membrane, there are molecules that do just that. They pass electrons down the chain. But as they do that, so notice NAD is going to give its electrons to the electron transport chain. As they do that, they create an ability to pump hydrogen ions across that membrane, which creates a gradient. And it does it for NADH, it does this 3 times, right? So it pumps hydrogen ions across 3 times. At the very end, those electrons are given to oxygen and it forms water. For FADH2, it only pumps 2 sets of hydrogens across. The difference is there that it's going to produce less ATP. Once these hydrogen ions are pumped across, then they're only let back in. And notice this is aerobic because oxygen is the final electron acceptor. So for prokaryotes, this happens on their cell membrane. For eukaryotes, this happens within the mitochondria, those membranes within the mitochondria. So here's the mitochondria. It's from the inside the matrix. It pumps the hydrogen ions into that space between the outer membrane. Very reminiscent of a gram-negative bacteria. Those hydrogen ions can only come back into the matrix through another protein port, which is an enzyme called ATP synthase. Because what it does is it adds the phosphates to ADP using the energy of the flow of the protons through that port, right? So it's almost like how water can turn a turbine. In this case, the flow of those ions rushing back to the other side creates the energy necessary to make that bond of adding another phosphate. Then when you take that phosphate back off later, you release that energy. This is called oxidative phosphorylation. And again, this has to do with the loss of electrons, right? Oxidation, loss of electrons. It's the electrons from the electron carrier, from the original glucose molecule you broke down, was what generates the energy that allows us to add the phosphates One thing that can happen if you have an electron transport chain, and especially if you're in the presence of oxygen, most organisms, as we know, either utilize oxygen or can tolerate it. When they have an electron transport chain, sometimes those electrons can jump ship. And when they do that, they can bind to oxygen in a way that creates a very reactive form of oxygen referred to as superoxide. This is bad oxygen. This reacts with molecules in a damaging way. The other thing that can happen is that in addition to the electrons, protons can be added and you can form hydrogen peroxide. Also a destructive form of oxygen, not quite as destructive as the superoxide radical, but still can be damaging to cells. So our catalase test is a test for a particular enzyme that has the ability to break down hydrogen peroxide into a safer form of oxygen again. It breaks down hydrogen peroxide, and this is the important reaction you need for your report. It breaks down it into water and gaseous oxygen. This is why you see what? Bubbles, right? Because the oxygen is in gas form. Those bubbles contain oxygen gas. Yeah, this top one, this complete equation. So the important thing is that what you guys did today, because your organisms contain catalase, they catalyze this reaction of turning hydrogen peroxide into water and gaseous oxygen. That's why you saw bubbling, because your organism has catalase. So do you, because you're aerobic, right? We live in an aerobic environment. We have an electron transport chain. We utilize oxygen. So this happens in our bodies. And we have to have a way of protecting ourselves. So breaking down, right, the catabolism of glucose gives you several electron carriers and generates, even at the substrate level, ATP or GTP. It's also going to form metabolic water, and the carbon is going to be released in the form of carbon dioxide. So again, we're removing electrons from carbon, and we're adding the electrons to the electron carriers, so they're being reduced. That's an oxidation-reduction reaction. On the flip side, right, as I said, we're generating some energy right at that enzymatic level. In the electron transport chain, 3 ATPs are produced per, because remember we said 3 pumps of hydrogen ions across per the carrier of NAD. At H, as opposed to FADH2, that one only does 2 pumps, you only get 2 ATPs. So therefore, aerobic respiration, glycolysis, right, the gateway step, the Krebs cycle, and the electron transport chain, all of that combined to form aerobic respiration, the yield could be 38 ATPs in bacteria. In eukaryotes, however, the yield is only 36, because it has to do the movement from the cytoplasm for glycolysis and then into the mitochondria, and so that we lose some energy in that process. 4 of the ATP, if you kind of lump in the GTP with it, happen right at the substrate level. 34 of that number comes directly from the electron carriers carrying those electrons to the electron transport chain, right, and producing ATP through oxidative, because of electrons, phosphorylation. So these are theoretical yields, right? In reality, though, it might be a little less, about 29 to 30 ATPs per molecule of glucose. So glucose plus oxygen will yield about 6 carbon dioxide molecules and about 6 molecules of metabolic water. Theoretically, about 2,000-ish kilojoules per molecule. Each ATP, when you break it down, you remove that phosphate, it will give you about 30 kilojoules per molecule of energy. Times that by 30, right? Notice that that's only about 1,000, so you only get about a third, or 33% of the energy return, but it's better than nothing, right? It clearly is effective. We're still alive. Organisms are still alive. There's two cool videos that I don't think I've added yet, but as you guys will notice, I've pretty much updated for the most of the semester our Canvas site. There's one that's an animation that shows you, because for some of you guys, seeing that stuff go makes sense. And then this one is a rapper singing about it. It's quite hysterical. So, if an organism lacks electron transport chain, how does it recycle the reduced coenzyme, this electron carrier, NADH, that you generate definitely in glycolysis? How do you do that? If you don't have an electron transport chain, can you do it? Or maybe you don't have oxygen, so you can't use your electron transport chain. What you will do instead is you will add those electrons back to your original product that you broke down, that glucose into pyruvate, and you're going to produce acids, alcohols, and or gases. We call this process fermentation. So we're going to add those electrons back to some organic molecule in the cell. So lots of pathways. By no means are we memorizing these. This is not biochemistry class, right? He's going, thank goodness I have that though, right? So just to give you some examples of what happens. So break down glucose by glycolysis. You end up with pyruvic acid. You can remove carbon dioxide and form formaldehyde. This can actually pick up the electrons from NADH that they picked up in glycolysis and form ethanol. This is one of our favorite products of fermentation, right? So this really nice 2-carbon alcohol called ethanol. Organisms that have this ability are called brewers or baker's yeast, right? This is our good old Saccharomyces that we looked at the very beginning of the semester. Another option is, again, remove carbon dioxide and form formaldehyde, but then actually add a pyruvate to it, and you form can't say these names. Acetolactate. There you go. 5-carbon, again, acid. That can later, you can add protons to it and release a carbon dioxide molecule and form acetonin. This is different from acetone, right, that us women are used to for removing nail polish, right? This is a different molecule. Acetonin then can pick up those electrons from the electron carrier NADH from the glycolysis that generated this pyruvic acid. And we can form a different alcohol called 2,3-butanol. And this one we tested for the presence of, and this is a rate limiting step going from here to here, in the Voges-Proskauer test. We actually test not for the 2,3-butanol, but the ability to make 2,3-butanol from the acetonin. So if the organism produced the acetonin, the reagents we used bind with the acetonin and produce that red color. So that's a particular type of fermentation, right? Being able to produce 2,3-butanol. If your organism was VP positive, it does that type of fermentation, breakdown of glucose. Another example is taking pyruvate and it picking up the electrons and forming lactic acid. Lactobacilli and sourdough bread, right, do this and produce that sour taste. Remember I mentioned before, acids taste sour to us. Lactobacilli in the human vagina lowers the pH, right, that acid type environment. Striated muscles during brief anaerobic periods when they don't get enough oxygen, right? They can't use their electron transport chain. They're still breaking down glucose for energy, but they produce that pyruvate. It can't go anywhere. You don't want to generate a whole bunch of electron carriers if you're not using your electron transport chain. So instead, we produce lactic acid. Electron carriers can lactic acid can pick up electrons from the electron carriers and produce propionic acid. No organisms have evolved to do it this way. They produce propionic acid a different way. They form oxalate, which remember is one of the intermediates in the Krebs cycle. They convert that into succinate by adding the electrons to that. These are 3 steps again in the Krebs cycle, but gone in reverse. Enzymes can go in multiple directions, some of them. Then they form propanate, right? This particular acid. And in fact, Peter's a stickler for it's not exactly pyruvate. It's actually a molecule very similar to it, but close enough for the purposes of this class. So again, pyruvic acid can be converted into acetyl-CoA, which could normally enter into the Krebs cycle. But, you know, different portions of it are going to generate different things. You're going to get lots of different acids, alcohols, or ketones. So this is an example of forming formic acid. No oxidation reduction reaction here, though. No electrons moving. But then that could, of course, go into the Krebs cycle. So your phenol red broth test detects both gas and acid production. So in the case here, notice carbon dioxide gas and hydrogen gas were produced by this form of fermentation. This test can also tell if the organism utilized the protein source by deaminating it. And that's when you get the pink result. Your methyl red test is a pH test as well. And this lets you know if a large amount of acids, right, mixed acid fermentation has happened. So anaerobic bacteria produce hydrogen gas. This causes bloating in dead animals. A common spore-forming anaerobe, Clostridium perfringens. This is one that will cause gas gangrene for living individuals, but this is usually the one that will cause serious problems and will cause the bloating of a dead animal because of the gas buildup from the organism growing from within. They literally can explode.
Whales.
Yeah. Beast whales. Early airships, right, also used hydrogen gas. The problem with hydrogen gas, too, is it's very flammable, right? So the most famous example of this is the Hindenburg explosion that happened in 1937. So now they don't use hydrogen, they use helium, which is lighter than air and not explosive. And, of course, fun to play with, too. So possible products of fermentation. It's going to depend on the organism and the environment they're in, right, and what type of sugar you give them. But they can produce, and the 1 stands for how many carbons are in these molecules. So a 1-carbon acid known as formate, or a 2-carbon acid known as acetate, or acetic acid. Propionate, lactate, butyrate, or succinate. Alcohols, ethanol, isopropanol, butanol, 2,3-butanol, and then, of course, methanol, which you definitely don't want to ingest. It's toxic. Ketones like acetone, Gases are either carbon dioxide, which is difficult to distinguish whether it was from fermentation or respiration, right? It's produced in both processes. But hydrogen gas is unique to fermentation. As I said, methanol is toxic. It can be produced from carbon dioxide or forget what that chemical name is for CH4. I think it's more than a methyl group. Is it methane? I think it is methane. By certain anaerobes, but this is toxic to aerobes like ourselves. Following oxidation to formate, because it inhibits cytochrome C. This is one of the electron carriers on our electron transport chain. So in humans, methanol poisoning can lead to permanent blindness and death because the human optic nerve, right, we're constantly always using this, right? It's constantly respiring, it's very active. And so when you shut that down and you cause damage to that nerve, you could permanently damage it so that you no longer can see. And then, of course, the brain, too, right? Very active constantly. So you don't want to kill that off. Don't be drinking any methanol. Okay. So, as I said, the methyl red and Voges-Proskauer test, we use the same media to run these tests, so we split it into two tubes. The mixed acid fermentation, which one of these tests detects mixed acids? It's the methyl red. What's the reagent that we use? Methyl red. And what methyl red is just a pH indicator. That's all it is. What do the products of this type of fermentation do to the pH of the media? Right? Acids are what? Low pH. Right? They lower the pH. So as I said, proton, aka hydrogen ions, just its proton, the smallest ions in all of chemistry, but probably perhaps the most important. And these are the ones that are factored in when we're measuring pH, right? These hydrogen ions, these protons, all by themselves. As we saw, they move around and their interaction are very important for chemical systems. So they're significantly important. So pH is actually the measure of hydrogen ions. P stands for partial pressure, right? H of hydrogen ions. So pH of around 7 is what is considered neutral. This is an inverse relationship. The highest concentration of hydrogen ions gives you the lowest pH. It's the negative log. And a low concentration of hydrogen ions gives you a high pH, what we refer to as basic or alkaline conditions. So, low pH, high hydrogen ion concentration. High pH, low hydrogen ion concentration. Did I say the other one backwards? I don't know. Let me say it again. High hydrogen ion concentration, low pH, acidic condition. Low hydrogen ion concentration, high pH, basic or alkaline situation. So there's lots of pH indicators out there. Some of them that we utilize are on this chart. So phenol red, as you'll notice, right, that it turns yellow below 7. So it helps you detect acids by turning yellow at that lower pH. Above that, it's going to be orange to that pretty pink color. Methyl red we use, and you'll notice it started out red, right? So it started out quite acidic down here in the 4 range, and it stayed red if your media was extremely acidic. It changed to yellow or orange if it wasn't acidic enough, if it wasn't 4.4 or lower. Bromenthal blue is what gives the citrate tubes their color. Around neutral, they're going to be green. When they become basic, they turn blue. So 2,3-butanol is which test? The Voges-Proskauer Test, right? Your VP test. What were the reagents? We used 2, didn't we? Barrett's reagent, A, alpha the naphthenol, and Barrett's reagent, B, which is potassium hydroxide. I promise I won't make you spell them on a test. But you do need to write them in your report. So these react with a particular chemical that's produced in 2,3-butanol fermentation. And what is that? Acetonin. Not acetone, acetonin. So as you can see in your book, as I said, your organisms took the glucose in the media, broke it down to pyruvate, but then could not utilize it in the Krebs cycle for one reason or another, converted it into alpha lactate, acetolactate, and then into acetonin, which then later can be converted into 2,3-butanol. When we added the alpha naphthenol, it bound with the acetonin. Then we added the potassium hydroxide. It also bound with these molecules. It forms diacetyl, which then binds with guanine, which is in the media itself, and it will produce a red color. So acetonin is what we're detecting. And remember, when we added the second reagent, I said really flick your tubes. And the reason for that is we needed to add oxygen into the liquid for this reaction to proceed. So notice oxygen is part of the equation.
So that's the ingredient that we're detecting?
Acetonin, yeah. So for both the VP and methyl red tests, what is positive? Red. For the methyl red test, it's because the pH indicator stays red, indicating it's a very low pH, around 4.4. For the VP test, right, it's because of that chemical reaction that happens. And this is why we had to wait a while, right? Time for that reaction to happen. So for each of these tests, each group is going to run negative and positive controls. And I've kind of eliminated the confusion of that. And I became control girl today, right? And I ran those controls, and you guys saw that. But why did we do that? Why did I run those controls?
Make sure everything was accurate.
Yeah, make sure everything is accurate, that the reagents are working appropriately. So I used known positive and negative organisms, right? And I had even pre-tested them yesterday to make sure that the reagents were good for you guys to use them today. So I knew it was going to come out good today. So we want to know what positive and negative results truly look like, and we want to make sure that our reagents are good. So that was just our procedure, what we did. All right, these are some examples of positive results. This one was a really weak result, and this one is that weird amberish color that sometimes you'll get that's not positive. It had to be a red result. And I know those are kind of pink, but it's within the red spectrum. Would a false negative result for the VP test be attributed to poor sensitivity or poor specificity of the system? Remember we talked about these kind of complications you can run into when you're testing things? So if it's negative, right, it didn't change color, right? So are we dealing with a specificity issue here? Are we being specific for what we're testing for? Yeah. But is there a certain limit to the sensitivity? Yeah, we have to be able to see that red color, right? So here it's a sensitivity issue. When you're looking for a color change, right, or be able to see something, this is usually a sensitivity issue. So if there's not enough acetonin to react, right, not enough to produce that red color that we would actually see it. What's up, Hunter? Okay. You're welcome. So again, it's quickly converted to the end product, and there'll be very little amounts of the intermediate product left to detect, if that was the case. But that's not the case in this case. Acetonin does stay around. It doesn't all get converted into the 2,3-butanol. There are other circumstances where it would be low. For instance, perhaps the acetonin lingers around for a long time in a particular species of bacteria, but not much acetone is generated in the first place due to a low amount of bacteria inoculating into your cultures. And these cultures have been going since like 2 weeks, right? So there definitely would have been enough acetonin in there for you guys to detect. At least there should have been. Whenever the concentration of the acetonin, for whatever reason, is below the limit of the detection of this test, right, this is when you'll end up with false negative results. So why were you told to shake the VP tubes after adding the reagents? One, to mix, but more importantly, to add the oxygen, which is required for this reaction to happen. So this is our protocol for methyl red test. Again, it's just a quick pH test. You put the reagent in, the pH indicator, and you read your results. This is one of my favorite ones, though, because I feel like a magician, right? Because for the negatives, the red goes away and it turns yellow, and the media is kind of yellowish. So unless you don't look closely, it almost looks like they disappeared, right? Yeah. Again, when you're doing pH tests, the color change just gives you a range of pH depending on that pH indicator, right? As you guys saw, I showed you all those different indicators, right? And their ranges of changes of color based on pH. So for the methyl red test, some organisms produce a mixture of acids, so much so that it becomes very acidic, right? For phenol red to stay red, it has to be 4.4 pH. That's a very low, very acidic pH. In addition, because of fermentation, right, they'll produce maybe alcohols and maybe gases, but this test is only detecting a lot of acid. Not telling you which acids, right, but the fact that they probably produce a lot. E. coli, for instance, under certain conditions will produce several acids: lactate, acetate, formate, succinate, in addition to ethanol, hydrogen, and carbon dioxide gas. He's a serious fermenter. So why is the methyl red test read immediately? It's just a pH test, right? But the Voges-Proskauer, right, you don't read it until at least 60 minutes have gone past, because what has to happen? A chemical reaction, right? And chemical reactions sometimes take time, right? Hence the waiting. Although you want things to go faster sometimes, it's just not possible. Although, as you saw, most of our organisms, within 20 minutes, they turn positive. So. And some as early as 10 minutes, we were able to see our results. Some microbiologists recommend re-incubating organisms producing a methyl red negative results for an additional 2 to 3 days. Why do you think this is done? What's going to happen over that longer incubation period? It's going to grow more, so therefore it's going to produce more. What are we measuring in this test? Acid. It's going to produce more acid, so therefore hopefully it ends up being that really low pH of 4.4. Typically these are incubated for 2 days. Yours have been incubating for 2 weeks. We shouldn't have had and didn't have any false negatives. If you're going to be positive, you were positive. No problem there. right? So the longer it grows, the more acid, the lower the pH, more likelihood you're going to get a true result. So why are we doing these tests? Right? We want to know what the organisms can or can't do. Can they do mixed acid fermentation? Do they have the ability to do 2,3-butanol fermentation? We properly run the test. We record our results. We interpret what these results mean. Right? So what does it mean if it's positive for methyl red? It means that it's a mixed acid fermenter, right? That it fermented the glucose, it produced a large amount of acid, lowering the pH to around 4.4 or lower. Hence why it stayed red when you added the pH indicator methyl red. These are the type of discussions, explanations you need to have for your organisms on your unknown report. Now, obviously, if it came out negative, you say it didn't do any of this, right? It's not a mixed acid fermenter. It did not produce a lot of acid from glucose. VP test, if it came out positive, you know your organism produced enough acetonin that you were able to detect it, that it can do 2,3-butanol fermentation. If it came out negative, it can't do this. So our phenol red broth. It's named this because it has the pH indicator phenol red. There's a particular way we code our results for this test. We use a base broth that has glucose already added to it. But as you can see by the manufacturer, there's a ton of different sugars you could order for testing the ability to ferment. Several different types of sugar sources out there. We, of course, are looking at glucose. So there is a protein source in these tubes. There's a pancreatic digest of casein, which is the protein in milk. Actually helps give it that white color. Pretty sustainable protein. The whey protein is the major component, though, in milk. In addition, salt for that isotonic balance. Glucose, of course, because we're testing to see if they can ferment that. Phenol red is the pH indicator. And then, of course, water. And it's adjusted to around neutral pH, which gives the media, you'll notice our uninoculated negative control in my demo rack is orange. So we have amino acid sources, we have carbon sources, two choices of food. So what are some common byproducts that we would see from pyruvate, right, or breaking down of glucose? Acids, alcohols, gases, right? Gases are hydrogen or carbon dioxide. You can't tell the difference, right? Unless we have a way of detecting. You just know it's gas. But it could be one or both of these gases. Which one of these cannot be detected by this test? Alcohols. We can tell if acid is produced, and we can also tell if gas, right? This is actually the only test we run we can detect gas production from fermentation because of that little tube inside that we call the derm tube that traps that gas. So what's the pH indicator? It's the name of the test. Phenol red. Phenol red at just below neutral pH, 6.8, so not very acidic, will turn yellow. At above 6.8, a little bit above neutral to basic, 8.2, it'll be red. Or what some people refer to as magenta when it goes up above 8.2. So this really bright pink color. So what color does the media turn if acidic products are generated? Acidic does what to the pH? Lowers it, right? So it's going to turn yellow. So if you have a yellow tube, you have an acidic environment, right? And we code that by writing A. A for acid. What color does the media turn if you alkaline product? So remember, alkaline pH goes up. In the case of this one, is going to be that pinkish magenta color. We code that K. Peter's got a question mark here because he doesn't think my pink is pink enough in the picture. I need to take a new picture, I guess. But can you guys see? I can see that that's pink, and that's orange. Or whatever you want to call it, magenta. What does a Dern tube help detect? Gas, right? So some of you guys had a bubble, an air bubble in here. That's a gas bubble. Either hydrogen or carbon dioxide or both or one or the other was produced. We code that by writing G for gas. And if you have a bubble, you should have drawn your tube and indicated that there's a bubble, right? If glucose is fermented, what happened to the pH? So if glucose is fermented, we produce what? Acid, which does what to the pH? Lowers it. So again, it's going to turn what color? Yellow. In this case, in this picture, notice no gas was produced. So we code that A minus, right, for negative for gas production. Yeah, there's different size bubbles. That's why you should indicate what size you think your bubble was. He had a huge bubble. Almost his entire tube is completely removed of media.
So small is considered negative.
No, small is small. Yeah. Okay, so peptone, which is a mixture of proteins and polypeptides, basically amino acids, are a component in the media. And some of the bacteria will deaminate that protein source and produce ammonia. Ammonia is going to raise the pH, right? And so hence we get the decrease in hydrogen ions, and you get that color change to that pinkish color. So carboxylation versus deamination of amino acids. So amino acids have a carboxyl group and they have an amino group. Decarboxylation is removing, of course, the carboxyl group, usually in the form of carbon dioxide. Deamination is when they remove the amino group from the amino acid. This can then be oxidized, right? Pick up electrons. When that happens, you form an alpha keto acid. And this is what sometimes will happen, especially for diabetics where they don't have glucose in their cells because the insulin's not telling them to take it up from the blood. And instead the cells feed on proteins, on amino acids. And when they do that, they produce these acids that can be toxic to the body. Not a good situation for us. So this one's showing an example of carboxylation, again, removing that carboxyl group. Oxidation, an example of oxidative deamination, again, removing that amino group and removing electrons. So when scientists conduct this test, sometimes they will use a base broth and a broth containing the sugar. The base broth just has protein in it. What you guys utilized has glucose and protein in it, a sugar and a protein. So what happens if they eat the glucose? They ferment it, right? So what are we going to see? Acid production. How do we know that acid was produced? The tube is what color? Yellow. And we know that that's an acidic pH, a low pH. What happens if your bacteria ate the protein? What happens to the pH? It goes up. And what color is it going to turn? That pink or magenta color. What happens if the bacteria eat all the sugar and then begin eating the protein? So if they eat the sugar, it turns what color? Yellow. When it starts eating the protein, it's going to go back up, right? So it's going to turn orange, and then if it goes really long, red or pink. A couple of you guys have one of those organisms, right, that if I don't stick it in the fridge, right, then it just keeps going, right, and it will eat up that sugar. It'll eat up all that sugar and then go right to the protein. So those organisms are kind of like me. I'll eat the candy while I'm cooking the hamburger because I just can't wait, right? Sugar is fast energy, right? Some organisms will use that very quickly, and then they're going to move on to the protein. Some of your organisms, right, were just straight up pink for you guys because they do not utilize the sugar in a fermentative way at all. They utilize the protein as a food source. So, but if you had a protein-only tube, if we used the base broth and had no sugar added to it, the only possibility that can happen is for them to eat the protein, right? So the only color possibility we could have is what? Pink. Yeah. Just pink. And that's, again, if they deaminate, they break down that protein. So when we're interpreting this table, if you have the base broth and glucose, remember it could ferment. It could ferment with gas production or without. It could eat the sugar, or it could eat the sugar and then go and eat the protein. Lots of options here, right? With the base broth, the only choice is eat the protein or don't do anything else. So when we're looking at this table very easily in this second column, if you see anything other than K, is that a reliable result? No. How do you get acid and gas production from proteins? You don't. Something went wrong. This is a problem, right? Remember, Ag stands for acid and gas production. That can happen with the sugar, right? That cannot happen with the protein. This would not be a reliable result. Could they choose to do nothing at all? Yeah. They could die. Not create any color change, you end up with an orange tube. Could they eat the sugar, ferment it, produce gas, and then in a protein-only environment, go ahead and eat that protein? Absolutely, right? They're like, okay, fine, right? All I have is protein, I'll eat it. That's a reliable result. Can they, in a protein-only tube, ferment that and produce acid? No, this is not a reliable result. How did that happen? There's no glucose there. In the sugar tube, even though there's sugar there, they just chose to eat the protein, right? You have a pink tube, you code it K. Of course, given just protein, they make the same decision. They're like, okay, I'll eat the protein. This is a reliable result, right? If given protein and they eat it, right? Given the choice, sugar or protein, they chose protein. And then they don't do anything in the protein-only tube. Why wouldn't they eat it? Something's wrong here, right? Given sugar and protein, they ferment it, no gas production. Given the choice of just protein, they eat the protein. Is this a reliable result?
Yes.
Yes. Ate the sugar, right? Acid, no gas. Somehow in a protein tube, it's yellow with a gas bubble. Is that possible? No. Right? So this would not be a reliable result.
Reliable to determine if phenol red is present.
No, phenol red is a pH indicator, right? Yeah. So if you're interpreting your results, right, and your controls come out like this, do you count your can your results be reliable?
Okay.
No, because your controls are not as they should be. Right? This would be a set of controls. And the important thing here is that your protein-only controls can only turn pink. And if your organism eats protein, it should eat it again. When in the sugar tubes, it has a choice, protein or sugar. Sometimes it'll choose sugar over protein, right? But it cannot produce acids or gas without sugar. In those protein-only tubes, if you are getting a sugar-type result, someone contaminated your tube with some sugar, right? And so therefore your experiment is crap and you got to start over. It's not the case for any of you guys' tubes, okay? Everything is as it should be. So suppose you inoculate with a slow-growing fermenter. So I'm telling you it can do what? Ferment. So it should at least produce acid, right? But you look at the tube, there's slight turbidity, which means it's growing, but you coat it minus, which means it's orange, right, and no gas bubble. There's no color change yet. Would this be a false positive or false negative?
False negative.
False negative, because I told you it is a fermenter, so it should be what? Positive. What's the problem here? Sensitivity, right? He's too slow. He's not producing enough acid for that pH indicator to change over. So early formulations of this had a small amount of carbohydrate, small amount of sugar. Occasionally they produced false alkaline pink results after 48 hours. Because the question we're asking with these tubes is, can it ferment the sugar? Right? And some of them, as we know, will eat that sugar first, like me, but then when they run out, they're going right straight to the protein. And when they do that, it's going to turn pink. And you're not going to see that it was once what? Yellow. Okay? So, what happened? But what happened? They ate all the sugar, right? There was a small amount. Within 48 hours, they blew through it. So, now what are at least 2 steps you could do to prevent this problem? Add sugar, which is what they've done. No? Because you want to know if it eats protein as opposed to ferment. But that's a good point. You can remove protein. The problem with that is that they still need proteins to live. So if you gave them just sugar, it's not going to be enough for them. But good argument. So you could add more sugar, and that's what they've done. But what else? In the incubator, shorter time, right? Catch them eating the sugar before they get to the protein, right? And again, what I had to do for some of your cultures, because you don't come in here 2 days after you inoculate them, right, is I put them in the refrigerator. Or in the case for some of you guys, I forgot, and so we had to do it again. So you could see, catch them eating that sugar before they moved on to the protein. So that you know it does have the ability to ferment that sugar. Because that's the main question we're asking. Right? We want to know, can they ferment glucose? Or, right, could they possibly use the protein as an energy source? We just inoculate the tubes, we incubate them for 48 hours at the correct temperatures, and then we look at our results. We code them as such, but what does this truly tell us? It tells us if the organism is capable or not capable of glucose fermentation. Right? We can detect this by the fact that there's a pH indicator that turns yellow when the pH lowers under acidic conditions. Catalate, not catalase, not yet. Citrate. Right? This test is actually testing for a protein embedded in the membrane of your bacterial cells that has the ability to transport citrate from the media into the cell. This protein is called citrate permease because it acts as a port. Now, of course, my picture is for sucrose, but, same principle. I couldn't find a one, right? But it's going to take sugar into the cell. for each of the sugars and everything that these cells process, they have to have something to transport it into the cell. So some of your bacteria have this. In this media is very evil. It only has one carbon source, and that is the sodium citrate. All organisms need a carbon source and a nitrogen source, and again, only one choice. Ammonium dihydrogen sulfate. Again, I promise I won't make you spell it or write it out on a test. Since this media only has one carbon and one nitrogen source, what will happen to the organisms if they cannot take in these molecules into their cells and use them?
They perish.
They die, right? So you'll notice those of you guys that have a green slant, you shouldn't have any growth. If you think you're seeing growth, it's probably a heavy inoculum, like you put too much bacteria on there to begin with. They didn't actually grow. And these are so old that you really shouldn't see anything at all. What's the pH indicator? Bromenthal blue. So this particular one, when it gets above neutral, when it's in the basic or alkaline range, it's going to turn blue. And this is because of them utilizing the ammonia source, producing the ammonia, from the nitrogen source, which they can't do if they're not utilizing the citrate, right? You need a carbon and you need a nitrogen. If you can't take in the citrate and use the carbon source, you won't take in the nitrogen source either. So these organisms are doing both. So decrease in hydrogen ions, increase in pH, turns blue. So as they consume the ammonia from the media, they deplete the hydrogen ions, pH goes up, media turns blue. If the media is green and you see growth of the organism in the tube, is this a positive result for the utilization of citrate? If it's truly growth, yes, right? But as I said, for you guys, it's not growth if you see something. It's probably an over-inoculation, which can create problems, right? False positives, you just went like that. Trust me, these have gone long enough. If yours is supposed to be blue, it's blue. So, again, they can only grow, truly grow, unless they have citrate permease. So again, your plates should be read the next day, but what I do is I take them out of the incubator and I stick them in the fridge to preserve them for when you guys come back. And I usually take pictures. Okay. So citrate positive organism might not produce a color change. This would be a false, is this a false negative result? Yeah, because if it's citrate positive, it should produce the color. What's the problem here? Right, not enough ammonia being utilized to change the pH. So what is this, sensitivity issue or specificity? This is a sensitivity issue again. You see the theme here, y'all? We actually do have one specificity we'll talk about. It's coming up. All right? So I'm not going to go over the scientific method, but we know this by now, right? Yeah. And you guys have it in the PowerPoint. So the last one, catalase. So flavoprotein is an electron carrier, of course, in the electron transport chain that passes electrons to oxygen. But sometimes this guy screws up, right? And he produces a very damaging form of oxygen called superoxide. So guess what we have? An enzyme that can convert superoxide radicals into a not so damaging form of toxic oxygen we know as hydrogen peroxide, and a safe form of oxygen. This enzyme is superoxide dismutase. So that takes superoxide, right, that charged, highly damaging form of oxygen, and converts it into hydrogen peroxide. which is still bad news, right? So the next step is what? Catalase. Catalase converts the hydrogen peroxide into water and gaseous oxygen. So hence you see what?
Bubbles.
Bubbles. Bubbles, bubbles, bubbles. So we did this on a microscope slide, right? Most of you guys saw it quite well. And we also did it right on our slant. And that tube is definitely dead and gone. Those organisms are not usable. With a false positive, and this is why it was important, right, that we put the bacteria on the slide first and then added the hydrogen peroxide. Because what instrument do you use to add the bacteria to your slide? Your loop. What is your loop made out of? Metal. Metals can cause this reaction to happen, to cause hydrogen peroxide to turn into water and gaseous oxygen. So we wouldn't know if we put the hydrogen peroxide on the slide and then added the bacteria, if it was the bacteria or the loop causing the bubbling, right? So this would be an example of poor specificity or sensitivity in this case. Specificity. And you really only run into that problem if you don't perform the test properly, right? If you do it right, no problem. So a weak catalase positive, which a lot of times will happen when you're doing it on the microscope slide because it's hard to visualize. Sometimes those bubbles are really hard to see. So you might not see them, so you record this as a false negative result. Is this poor specificity or sensitivity? Sensitivity, right? It's difficult for you to visualize. That's a sensitivity issue. Maybe viewing it under the microscope would help. But we do the slant test with lots of bacteria, right? As a backup. Hydrogen peroxide is the reagent we use, right? Because we're testing to see if you have the enzyme catalase. Because what does catalase convert hydrogen peroxide into?
Water.
Water and gaseous oxygen. That's why you get the bubbling. So why was we advised and why did I do as a control a catalase positive organism? What are we checking? Make sure the hydrogen peroxide is good. Notice it's kept in a dark bottle. Light can cause this reaction to happen. Over time, right, your hydrogen peroxide turns into water and gaseous oxygen, so the oxygen, leaks out of the container. It's also why I want to keep it sealed tight. So a lot of people used to use this as an antiseptic, put it on your skin. There's an old wives' tale about that if it doesn't bubble, it's not infected. That's bullshit, right? It should always bubble, because guess what? You're catalase positive. You're an anaerobe, right? Do you produce hydrogen peroxide? Yeah, you do. So it should always bubble. If it doesn't bubble, it means you just poured water on yourself. Okay? But like for women and those of us, right, we take our earrings out, you can put it in hydrogen, it's a really good cleaner, you can use it to clean your home. Although nowadays, because of the damaging effects to even your own cells, they suggest that you don't use this as an antiseptic anymore, right? So, you can clean your earrings, right, your you name it, right? They put it in your detergents nowadays because that bubbling action helps in breaking things up, right? And it will also kill bacteria. The problem is it will damage your own cells too. You don't want to drink it, right? You can rinse your mouth with it, but it's nasty, right? But they say soap and water and good old triple antibiotic ointment for your cuts and scratches. So what's the purpose of adding it to the uninoculated tube? Why did I do that? It was a control to make sure that the media itself does not cause that reaction either, and it did not, right? So it's safe to go ahead and just put your hydrogen peroxide right on your slant, and if it bubbles, it's your organism, not the media, causing that reaction. So that was a negative control. So our good old scientific method. I wasn't kidding when I said today was a long one. Aren't you guys glad I came in early and set up all your stuff for you? So this is the Enero tube. This is the last thing. Not anything you'll be tested on. Right? You can read about it in your book. These chambers, each one of them run a different test. Some of these look familiar to us, right? A lot of them are sugar fermentation tests using phenol red. So they turn from red to yellow if they ferment the sugar. There's even the good old citrate test, which we did, right? Goes from green to blue. There's other cool tests. The VP test can be performed by adding the reagents to the chamber. This is Taquana's favorite one. It produces a magenta positive color result. But in a couple of other tests, these have wax over the chambers so that it's done in an anaerobic environment. And so especially if they ferment glucose and produce gas, it'll actually pop the wax. And like I said, I had some that showed this before, but I've lost them.